Hello class, here's the second section of the digestive system. We'll again be going a little bit more into the physiology and some of the digestive process as we're entering into the stomach area of the stomach, and I think also the intestinal, small intestines that we'll be talking about in the next section of the chapter. So we can see again, stomach, we saw in lab, again, its shape depends on, again, how much food is filled. It can expand to about 1.5 liters of material. Again, when that material is digested, we call it chyme. So the viscous, acidic, soupy mixture of all the food, saliva, and material in there is referred to as chyme. And this is also why we wouldn't be doing a lot of starch digestion in the stomach, because even though the saliva is ending up in the stomach with our food, the acidic nature of the stomach is not suitable for that salivary amylase to function very well. So we're not going to be doing a lot of starch or lipid digestion in the stomach just because of the acidic nature precludes the enzymes from functioning well in the stomach. But there are some protein enzymes the stomach secretes that can aid in the digestion of our food. The acid doesn't digest our food, but it can help denature proteins, kill some of the bacteria in the food that we're eating, but doesn't really digest, because again, chemically digesting is breaking down. You might say the acid might be involved in the mechanical breakdown of the food along with the churning process, but again, it's not chemically digesting our food, right? There's some curvatures to the stomach, the lesser and greater curvatures we saw in the lab. In the beginning of the stomach, where the esophagus meets, is usually referred to as the cardiac region of the stomach. This enlarged area above it is called the fundus area. The major part of the stomach is called the body, and then as it tapers down and makes that last turn towards the intestines, it's the pyloric region of the stomach. So again, these are just functional regions we saw in the lab. And again, this is look at the stomach underneath the liver, where we can see the gallbladder associated. Again, the ducts that would take the bile away from the gallbladder towards the stomach. You can see that greater augmentum, that fatty layer of membrane that hangs down the stomach. Now, while we mentioned in lab and saw in models in the lab and mentioned earlier in this lecture that the intestinal tract has two layers of muscle, a circular and a longitudinal layer, here the stomach has also an oblique layer, right? So we got that longitudinal layer that would be running the direction of the stomach contents, the circular layer that's a band around the stomach, again, going a circle around, and then we have a muscle that goes at a 45-degree angle, sort of in between the two directions of the longitudinal and the circular muscle. So it's a third layer of muscle. Again, the stomach is doing a very important job of mixing, sort of pummeling that food as you're beginning the really significantly beginning chemical digestion. Right? It's aiding in the mechanical digestion of our food. Theoretically, again, we did a significant amount of mechanical digestion while chewing the food, but we don't always completely break down the food with our teeth mechanically. So the stomach, with this powerful layers of muscles, are going to churn and mix up that food to really break down the components mechanically with, again, the aid of the enzymes. right? So those are the layers. Again, the stomach mucosa layer has these rugae. Again, the rugae are created by folds in the muscle. So when the muscle layers are more relaxed, creates ridges and folds along the intestinal mucosal layer. And again, when we stretch the stomach full of food, those rugae would flatten out, again, allowing the intestinal or the stomach epithelia to stretch along with the stomach as we're filling it with food. Right? Again, you get that pyloric atrium, the beginning of the pyloric area from the body, and then you get the pyloric canal. That's just meeting the duodenum, beginning of the intestines. Again, we have a very strong muscle, the pyloric sphincter at the end of the stomach, much stronger than the gastroesophageal sphincter at the beginning of the stomach with the esophagus. This is really going to help hold in the food into the stomach until it's been properly digested by the activity of the stomach, so that when we're releasing it into the intestines, it's ready to be further enzymatically digested by the enzymes of the intestines and the pancreas we'll talk about. Right? Pyloric sphincter. Again, a powerful smooth muscle in that junction between the stomach and the duodenum. So again, stomach is just a temporary area to hold the food while we, again, do more mechanical digestion. Acid kills some of the bacteria, not all, but some of the bacteria in our food. It can help denature proteins in our food, which again can be part of sort of that mechanical digestion of the food while the stomach is churning the food. So again, we're just mechanically breaking down the food, but not breaking the bonds in the monomers to create, again, a chemical digestion. The only real enzyme that's in the stomach for digesting proteins is an enzyme called pepsin. And again, the stomach, as we mentioned in the blood chapter, could produce a factor called intrinsic factor that's necessary for vitamin B12 absorption and helps with, again, production of red blood cells. Lack of intrinsic factor would mean I wouldn't absorb B12 and have that condition we call pernicious anemia in the blood chapter. So again, this is why A and P, we can't just sort of talk about one organ system by itself. They're, again, interconnected. Right? So the mucosa layer, again, we looked at some of this in the lab histologically. Right, so we have these simple columnar epithelia cells with a lot of mucus-secreting goblet cells. Right, lifespan of an epithelia cell in the stomach is 3 to 7 days. So we're regularly replacing all the cells along the epithelia layer of the stomach. Right, because they're getting a lot of damage from all of the digestive process. We're not digesting away the cells, but again, they're getting a lot of damage from that activity. So the cells are going to be highly capable of doing mitosis, dividing, producing new cells, and growing new cells, and replacing damaged cells as they're dying along the area to help maintain the epithelial boundary. Again, creating these gastric pits. So these are going to be sort of downwards projecting folds in the lining of the stomach versus, again, projecting outwards. These are, again, more what we call gastric pits. Right? So again, submucosal layer still has the blood vessels, lymphatic vessels, nerves pass through there. We got, again, 3 layers of muscle, and then we have that serosa layer. Right, the gastric pits have 2 major cells in the gastric pits. We have the parietal cells and the chief cells. These are, again, producing the gastric juices. So again, they're producing the acid in the enzyme for digesting our food. We also again have those enteroendocrine cells, these hormone secreting cells that can again coordinate the activity of the digestive system, right? So these gastric pits can be shallow depressions, again depending on the area of the gastric area, or some are more deeper. Each of them have again a large number of mucus secreting cells at the top to help protect the cells against the acid of the stomach, but again they have a large amount of these digestive secreting cell, or the gastric fluid secreting cells of those parietal cells and the chief cells, right? Each pit communicates with several glands within the area. So again, here is looking at the gastric pit. So you got these mucus epithelia cells at the top. There are going to be quite a few mucus secreting cells in that area. Again, we got our parietal cells, right, that are in there. We got our chief cells that are in there, and again we got these they're calling G cells. These that again can be referred to as enteroendocrine cells. These enteroendocrine cells produce a major hormone, the digestive system, called gastrin. Produce some other factors as well, but the major factor they're making in the stomach is a hormone called gastrin. So let's look at what each of these cells do. Those parietal cells, right, the ones that are in sort of that light orange color in this picture, are producing that intrinsic factor necessary for B12 absorption. But they're also the acid-producing cells of the stomach, producing hydrochloric acid, which we'll talk about how that works. Hydrochloric acid is again necessary for pepsin activation, the enzyme that again does protein digestion. So pepsinogen, as we'll talk about in the next one, we go over the chief cells, produces an inactive enzyme called pepsinogen. Acid activates that. The parietal cells will keep the acid content of the stomach at around a pH of 1.52. So very acidic. Remember, these are tenfold differences in the amount of acid, amount of hydrogen, when we're going from 7 to 6, 6 to 5. So you get all the way down to 2. It's a lot of hydrogen ions in that fluid compared to plain water. Right? The G cells, again, are producing a variety of hormones. The major one, gastrin, and there that are important for regulating digestive system activity. Chief cells, again, produce a protein called pepsinogen, an inactive protein digesting enzyme. Right? Remember when we went over cells in AP1, cells are made up of phospholipid membranes and lots of proteins along the membranes, and a lot of proteins within the cells as well too, right? So when these chief cells are producing this protein through the ER and Golgi, right, they're producing the protein as an inactive enzyme, pepsinogen. That pepsinogen, when released through the exocytosis process, would be activated when it comes in contact with acid of the stomach, and it gets cleaved into a protein we call pepsin, which again is now a protein digesting enzyme. These cells in newborns can produce an enzyme called renin, which is necessary for milk digestion in newborns. Since again, newborn babies are primarily, right, through evolution designed to feed on breast milk. Not that all babies are fed breast milk, but the formula simulates breast milk components. So their stomachs are designed to help digest the milk proteins and are necessary. Again, and renin would also be something you would use often in the production of many cheeses. So many cheeses, you're using this rennin material to help break down and produce the material you need for producing cheeses. Also, again, gastric lipase is produced, right? That can help with lipid metabolism. But again, that would be more in newborns, because newborns have a more or less acidic stomach, not as high acid producing. That's why babies can't digest food the way we do. And also, again, that lower acid content is going to allow a lot of some of the good bacteria initially that they're interacting with to colonize their stomach, creating what we again talk about microbiology as the microbiota, or the normal flora of your digestive system. But as adults, we're not making much gastric lipase, and even if we do, again, the very acidic nature of our stomach would inhibit any real gastric lipase activity going on. Hydrochloric acid production. How do we do that in our the parietal cells, right? They produce hydrogen ions and chloride ions and secrete them separately into the lumen of those gastric pits. How do we do that? Same way that our red blood cells did some of these activities, right? Our cells are doing cellular metabolism. They're producing carbon dioxide. That carbon dioxide, when it reacts with water in the cells, can produce carbonic acid, right? That carbonic anhydrase enzyme that we said in red blood cells would be in these parietal cells, convert carbon dioxide into carbonic acid. Carbonic acid dissociates into the bicarbonate ion and hydrogen ion. So CO2, carbonic acid from carbonic anhydrase. We produce bicarbonate ion, a buffering ion, hydrogen ions. We have a proton pump. This yellow structure is an active proton pump that's going to pump hydrogen ions into the lumen of the gastric pits. Bicarbonate ions are pumped into the bloodstream, and we pump chloride ions into the parietal cells that can then be transported into the lumen, because they will follow the positive charge and move in there, and that's going to create the hydrochloric acid in our stomach. Again, this is referred to the alkaline type because we're doing pumping of bicarbonate ions into the bloodstream, which again is a pH buffering component, right? Drugs that we use to treat people with GERD or peptic ulcer disease, or again, people who just have a lot of acid burning in their stomach that maybe don't have complete ulcer condition, but again, or maybe need to deal with that from their being stimulated too much from their diet. You can do various things. You can take something like Prilosec or any of the new versions of those. Those are called proton pump blockers. So this yellow circle here is a proton pump because hydrogen ions are protons. So the proton pump blockers like the Prilosec and Prevacid, I believe is also in that group, are proton pump blockers. They're going to block the pumping of hydrogen ions on. So if I take that, it will help neutralize or prevent acid building up in my stomach, make my stomach less acidic, and so I don't have as much irritation in the stomach. So that will help me out. Again, I'm not going to bring my stomach all the way up to neutral pH, but it'll be less acidic, so I'll be less bothered by the pH. This is a better approach than taking Tums or Rolaids, because if I take Tums and Rolaids, the Tums neutralizes the hydrogen ion, raising the pH of my stomach. But when the pH of my stomach is elevated, we said we had chemoreceptors earlier in this chapter that recognize a higher pH of the stomach that stimulate gastric cells to produce more hydrogen ions. So if I take Tums Rolaids and my pH goes up, my stomach just makes more acid to bring the pH of the stomach back down, which means I'd have more burning sensation. I would need to take more Tums to neutralize that acid, and I'm going to be on this yo-yo going up and down, constantly taking Rolaids and Tums to try to control this acid levels in my stomach, because whenever I try to neutralize it, I just make more acid. So blocking the pump would be more effective, because even if I stimulate the cells to produce acid, the pump is blocked and I can't produce or release the acid into the lumen of the stomach to lower the pH of the stomach. Now, before we had Prilosec and other types of proton pump blocker medications, there were earlier drugs like Zantac and Tegament and other ones that worked on the stimulation of the cells. What would stimulate a parietal cells to release acid into the stomach? First of all, that gastrin hormone we talked about would produce a stimulation to increase acid production. That parasympathetic nervous system pathway that we said stimulates digestion, so acetylcholine from the parasympathetic nervous system would cause more acid release from the parietal cells, along with histamine. Histamine is in our body and has functions outside of our immune system. So histamine would stimulate parietal cells to produce acid as well. Stimulate our parietal cells with histamine, acetylcholine, and gastrin. High levels of acid are produced, bringing the pH down. Now you might think that those early drugs were maybe targeting the effect of gastrin in the stomach, because that's the most specific to the digestive system, but those drugs were actually histamine blockers. But you may not be aware of, there are two major different histamine receptors, right? Your Benadryl and drugs like that are going to target your histamine 1 receptors for your allergies and stuff like that. The H2 histamine receptors are in this digestive system, along with other effects in the body. So taking that could block histamine effects on the cells, and did decrease acid levels in the stomach to help people manage their indigestion and their high levels of heartburn. But the other drugs were preferred, became the norm for treating drugs. They're a lot more expensive than the older drugs, but again became the preferred way to treat people that had chronic acid problems with their stomach. So again, the bicarbonate ions injected into the interstitial fluid of the parietal cells, right, absorbed by the blood vessels. Blood vessels again would move chloride ions from the blood into the parietal cells. That chloride ion could then be transported into the lumen of the stomach to create the hydrochloric acid, right? So if gastric glands are very active, the monobicarbonate ion released through them increases the pH of your blood, right? Sudden influx of bicarbonate ions is called the alkaline tide. And that's another reason why taking Tums and Rolaids isn't a great solution to chronic acid problems, because if I'm constantly neutralizing the acid in my stomach with that kind of drug, as we said, that triggers chemoreceptors to stimulate acid production. So I start pumping more acid into my stomach. As I pump large amounts of acid to neutralize or to deal with all that rolades, I produce all those bicarbonate ions and start pumping large amounts of bicarbonate ions into my blood that can again cause my blood to go into a more alkaline condition. Again, as we said in the respiratory chapter, more often people are going to be in acidosis than alkalosis. So this is not likely to create a severe problem unless, again, you're really taking large amounts of Tums all the time. So could start having some minor effects on your blood pH. But again, your respiratory centers could try, as well as the renal centers, as we will talk about, to try to regulate blood pH by trying to regulate how much carbon dioxide we're getting out of their lungs, as well as how much bicarbonate ions we might remove from the digestive or from our bloodstream and move into the renal system to excrete out of the body. Right? So this hydrochloric acid is necessary for our digestion process. Right? Circular folds in the intestines increase the surface area of the intestines. These used to be called the plicae circulares. These circular folds are the ridges along the intestinal epithelia. These folds again create a large amount of surface area in the intestines. So again, roughly 800 folds in the small intestine, mostly in the jejunum, where again we're going to be doing lots of food absorption. Duodenum's only a small part of the intestines, right? About 12 inches, foot or so. So again, that's where we're more doing digestion activities, beginning release of all the enzymes and helping in the starting of or completing the digestive process of our food. The jejunum, very important for nutrient absorption. Again, the food is moving through the intestines at a relatively rapid pace through that peristaltic activity, and that's going to again give us basically one shot at absorbing our nutrients from our food. So more surface area, better absorption of our food. And then on those circular folds and the other flatter surfaces of the intestines, you have those small finger-like projections, the villi we talked about at the first part of this section of the chapter, right? So again, more surface areas. You got circular folds, and then the entire epithelium of the intestines has the villi, these small finger-like projections, which again increases a lot more surface area. And then all of the columnar epithelia cells of the intestines have microvilli on them, a large amount of surface area. So we said in AP1 in the cell chapter that microvilli help to increase the surface area of a cell for absorption. So all of these give a large amount of surface area to the intestines. We said the respiratory system had probably about 30 times the surface area of your skin. Your small intestines is probably going to have somewhere around 200 times the surface area of your skin because of all of the circular folds, the villi structures, the microvilli on the epithelial cells gives a huge surface area of the intestines for nutrient absorption, right? So each villi would have a lacteal, again, blood vessel flow in there, again, in that area, as I said earlier, in that tissue just below the villi, and that mucus layer right above that mucosal muscularis mucosa, right? You could have some lymphatic nodules, some of that MALT, that mucosa-associated lymphatic tissue. There can be some small little crypts of intestinal glands activity. We don't secrete huge amounts of enzymes into the intestines from these, but we do produce some intestinal fluid that does have a little bit of enzymatic activity. It also produces some mucus. Right, so if we look at these intestinal glands, right, the Paneth cells, again, at the base of them, have roles in innate immunity, right, and again, producing defensin chemicals, lysozyme. So again, as we said, there are lots of innate proteins that can cause lysis of cells and attack cells. Again, part of the innate immunity. So these are, again, part of that response. So again, here's looking at the villus. We saw a model of this in the lab where we had lacteal blood vessels. Again, we saw this in the lab a little bit, where you could see all of these columnar epithelia cells covering up our intestinal with some of these goblet mucus secreting cells along there, right? So the villus is, again, going to help bring fat absorption right into the lymphatic system. Blood vessels are, again, going to be carrying nutrients away from the intestines and bringing that to the liver, right? Remember, in the blood vessel part of our lab where we learned all the names of the blood vessel, you had the hepatic portal vein circulation, taking blood away from the stomach, the intestines. That was, again, oxygen poor blood, but nutrient rich, and other chemicals absorbed through the digestive system to the liver to be processed first before being brought to general circulation. Right? So lymphatic lacteal, right? Again, lacteal comes from milky, because, again, lactose sugar, right? Lact is often a word we use to refer to milk, right? Transports nutrients that can't be carried in our blood because, again, of the high fat. They need to be carried by proteins that can't easily dissolve into our blood plasma. So this is important for fatty acid and absorption, where, again, these fatty acids, which are, again, fairly hydrophobic, only weakly hydrophilic, are going to be bound to many proteins, creating these what are called chylomicrons, these little tiny packages of proteins carrying some lipids away from the digestive system. Right? You still got that muscularis mucosa layer that, again, that is underneath the villi, going to help, again, contract and move the villi a little bit along the intestinal epithelium to, again, help move contents along there for absorption. It also can squeeze the lacteal as well to help push fluid through that lymphatic vessels that, again, doesn't have a pump, and as we said, in our lymphatic system, they have a lot of valves, and muscle contraction can push fluid through these, just like it can help push fluid through our blood vessels. All right, so here's histologically what the villus looks like. Again, the cells along the villus with these epithelial cells can secrete what we call brush border enzymes. These brush border enzymes are often the enzymes we need to finish digestive process. So when I'm breaking starch down from pancreatic amylase, that breaks starch down to a disaccharide called maltose. The milk sugar, lactose, disaccharide. When I break down proteins into short amino acid stretches that haven't been completely broken down to single amino acids, these brush border enzymes are going to break down those last few bonds and make things as easy to absorb by breaking them down into their monomer forms of the monosaccharides, amino acids, again helping to break down nucleotides in our food, because again, if you're eating real food, that food should have animal cells or plant cells in there that have, again, DNA and RNA in them. So these are the regions of the small intestines that we talked about in lab, so we want to again go over them here, but this is where most of our nutrient absorption occurs, as we mentioned in lab. Stomach not really designed for absorption. You can do a little bit there, a little bit in the large intestines, but most of the nutrient absorption is in the small intestines, a little bit, 10% in the large intestines. All right. So again, going back to that tic-tac-toe region of the body we learned in AP1, we can see the digestive system is throughout the tic-tac-toe region. So again, if you're having an issue with your digestive system, you might have discomfort in various regions, but it might be localized. Like if I had appendix pain, you would find that on the right inguinal region, because that's where the appendix is, right? Versus again above the intestines. Again, we have our liver there, so the liver again is going to be more in that right hypochondriatic region of the body, where again you might again have some discomfort in that area, going into the lower region right below it as well. So here's the duodenum, right, beginning of the intestines. This was again also where more mixing can occur, because this is going to receive that watery kind from the stomach, but it's not completely digested. Again, we have begun a little bit of starch digestion in the mouth, done some protein digestion in the stomach, but here we're going to receive that mechanically broken down food, partially chemically digested food, and start breaking it down with pancreatic secretions, the gallbladder, which again is associated with the liver secretions, to help digest our fat and other components of our food. Also a lot of that bicarbonate ions being produced in here that can help neutralize the acid of the stomach, right? Second part of the small intestines is the jejunum. This is the largest part of the intestines, with all those circular folds that we talked about to increase surface area and nutrient absorption. Ileum, right? Final segment, right? About 11 feet, right? Versus, like, I guess the ileum is a little bit bigger, right? 8 feet for the jejunum. Ileum is 8 feet, 11 feet here. So again, these two are the two major parts of that small intestines. Duodenum's the smallest, right? Again, still has some of those circular folds to help with nutrient absorption. Again, we also do a lot of water absorption. So all the water we're drinking is a lot of it being absorbed in the small intestines, while the next segment, the large intestine, focuses on water absorption, but it's only absorbing the last bit of water that we didn't absorb through the small intestines, right? So again, this is the three regions of the small intestines, right? So multiple hormones regulate our digestive system activities, right? Just like the stomach, these are very important hormones for regulating the intestinal phase of our digestive process, as well as potentially inhibiting the gastric phase of our digestive process. Right? Hormone gastrin, that was also again produced by those gastric pits, right? Gastrin secreted by the G cells of the pyloric atrium. These enteroendocrine cells there are important in regulating the digestive function of the stomach. Secretin, right? Secreted by chyme entering the duodenum. So as we release that acidic chyme into the duodenum, and those cells are detecting the acid of the chyme, secretin is produced, stimulates again bile from the gallbladder activity to start being produced, right? Gastric inhibitory peptide, right? GIP, right? This is going to be secreted when fats and carbohydrates, especially glucose, the complete breakdown of starch into glucose, enters the small intestines and inhibits gastric activity. So when we see that we have completely had a very effective mechanism of digesting our food, and we have again a lot of those monomers starting to appear in the food, we can inhibit gastric activity and stimulate pancreatic activity that can cause insulin to get produced by the pancreas to again get ready to help with sugar levels that are going to be brought into the body with all that glucose, right? So once we absorb that glucose, the insulin is going to be ready in the body to help transport glucose from our bloodstream into our tissues of our body. Cholecystokinin, CCK, important hormone, major stimulator of gallbladder activity to secrete bile from the gallbladder into the duodenum, along with pancreatic activity, going to help relax the sphincter at the end of the bile and pancreatic duct, to also again help with entrance of these fluids into the into the intestinal area. Vasoactive intestinal peptide, VIP. Lots of three-letter abbreviations or more in some cases, right? So stimulates again secretion of intestinal glands. So again, we have some of those intestinal glands in those pits. Again, dilates capillaries, inhibits again acid reduction of the stomach, right? So this is going to again help with nutrient absorption and also as well as inhibiting gastric activity, because as we move into the intestinal phase, we don't need as much gastric activity. We're still going to release chyme from the stomach into the intestines, but we don't need to again secrete large amounts of acid into the stomach because we've completed the gastric phase and now moving into the intestinal phase, right? The enteroendocrine also released by these enteroendocrine cells, again in response to stimulation of the vagus nerve, right? Can before the chyme reaches the pyloric sphincter, again helps with chyme entering the duodenum, right? So this is, and also help producing an alkaline mucus that can again help protect the intestines against the high acid level entering it from the stomach. So you can see there are lots of important hormonal components of the intestines that are important for aiding in digestion, right? So gastrin primarily produced within the stomach, right? Pyloric area, going to again stimulate acid and gastric muscle activity for digestion. That GIP, the gastrin inhibitory protein, right, would again inhibit gastrin activity, but also again stimulate the pancreas activity along with secretin, going to help promote activities of the pancreas, which include both insulin production and release of some of the digestive enzymes of the pancreas. CCK, again, along with secretin, are going to help stimulate the that activity as well. CCK, again, definitely targeting that gallbladder to help release bile from the gallbladder. And bile is an important, not enzyme, but emulsifying material for our fat. So bile will have bilirubin in it, but also has bile salts in it, which is important for helping in fat emulsification. So you can see all of these activities are going to be important for regulating various steps of the intestinal phase, as well as potentially inhibiting gastric phase activity. So again, I think I jumped the gun a little bit talking about phases of digestion. Right, so cephalic phase begins even before food enters my mouth. So just smelling, seeing the food, tasting the food, that higher brain function of the cerebral cortex, seeing and smelling, gets you excited about food, and that excitement will again help start to begin the process through the vagus nerve of stimulating my digestive system to prepare the stomach. So we'll again start producing some gastric juices and get the stomach ready for food digestion when we finally eat the food. Right? So cephalic phase, gastric production increases, right, to about 500 mLs per hour, right? Only lasts usually a few minutes, because again, typically we might only see our food for a short period of time before we eat it, unless again we're maybe in the process of making a very large dinner, maybe seeing the food for an extended period of time while we're actually preparing it. Once the food enters into the digestive system, we're in the gastric phase, right? So distension, stretching of the stomach wall, increasing the pH, because we're neutralizing the acid with all those components in our food, right? The acid is absorbed into those food components, and it basically neutralizes the acid, just like Tums can. That's why often if you have a lot of acid in your stomach, if you eat food, that can help the stomach acid be neutralized, so you don't have so much burning sensation in your stomach, right? Presence of undigested food proteins and materials stimulates the chemoreceptors to again create more gastric juices and motility to begin the gastric phase of digestion of our food. We're not going to last 3 or 4 hours while we're digesting our food into that chyme material. Right, so that's going to involve all of those cells and factors we talked about earlier. Once the gastric phase has digested our food, or at least mechanically digested our food and partially chemically digested our food, the factors can cause the pyloric sphincter to relax, and that again will allow chyme to start entering into the duodenum. And again, if we sense our chyme has been efficiently digested, we can again stimulate more relaxing of the pyloric sphincter and allow more chyme to enter into there. But again, so when the intestines detects all of these components entering the intestines, we're now entering the intestinal phase. All of those factors, the CCK, the GIP, the secretin, all of these things are going to stimulate gallbladder, pancreatic activity, but also again block gastric activity. Right? So we're going to inhibit gastric production, decrease gastric motility, stimulate pyloric sphincter relaxing, so more chyme can enter into the intestines, right? Gastric central reflex, again, is triggered by stimulation of the stretch receptors in the stomach wall. So again, while I've expanded my food or stomach with all the food I've eaten, that increases again all of the gastric activities, but it'll also get the intestines ready as we start dumping chyme into the intestines, so that we can again mix that chyme up with those digestive enzymes, so the pancreas and the bile, to help with nutrient digestion, because again we still got to do more nutrient digestion in the small intestines. All right, so those are all important parts of the small. So the large intestines is the end, or the last major segment of the alimentary canal as we leave the small intestines, right? It begins with a small part called the cecum, goes through the colon, which again is broken down to various regions as it moves along the abdominal pelvic cavity, ending in the rectum, that leads again to the anal canal for passing bowel movements out of the body. Right? Major function is the reabsorption of the last bit of water in our food. Not all the water, but the last remaining available water, because again, if we absorb all the water out of our food and create something that's too dry, it'd be hard to pass that through the bowel movements. But it's important for reabsorbing the water that is not being absorbed by the small intestines. Some vitamin absorption occurs in the large intestines from bacteria that exist in the intestines. So bacteria that are going to be digesting some of the plant fibers and nutrients, or the leftover of our nutrients that we couldn't digest and absorb. They're going to be breaking that down, but also producing some vitamins. Bacteria, as we talk about in microclass, can produce the some of the B vitamins and vitamin K. So those are important for, again, our overall health. Not huge amounts of vitamins, but enough to help supplement our activity. Right? Going to store fecal material at the end of the system until we're ready to have a bowel movement. Right, so here is the end of the ileum, the ileocecal valve, as it's referred to. Again, it's not so much a sphincter, it's a valve to help allow material leave the small intestines, enter the large intestines. So the beginning part of that is the cecum with the appendix, lymphatic tissue in there. Right, as you go up the cecum, we have what's called the ascending, going up colon, transverse colon, going across the abdominal cavity, descending colon, going down, a little twist to go posteriorly, the sigmoid colon that ends in the rectum, where we're going to store that fecal matter until we're ready to have a bowel movement. There's an additional layer of muscle in the large intestines called the tenae coli. So this is again sort of a band of muscle that follows the length of the large intestines, that creates these haustra pouches. So the tightening of that muscle pulls the intestines inwards and creates these haustras. There are also these little fat appendages along the intestines. Don't seem to have any real functional role of anything, those little fat appendages. So here's the cecum, again, where food is entering from the ileum, and we begin to again do the water absorption and compacting this material down as it moves through the entire large intestine area. Right, appendix attached to the area again has lymphatic tissue associated with their nodules of that. Again, some studies also say that another function of the appendix is to help store some normal, healthy bacteria of the intestines in there. So in parts of the world where very watery diarrheas called dysentery occur and you flush out the intestinal system, the bacteria of the appendix can help reseed the large intestines with, again, healthy, normal bacteria. Not that the dysentery will get rid of all the bacteria in the intestines, but this can help with adding back some of that healthy, normal flora back to the intestinal tract. But if bad bacteria or some other material gets lodged in the appendix, can cause inflammation of the appendix, and then it has to be removed because that bursts. It would release bacteria from the inner content of the large intestines into the outside, that serosal membrane, that would again cause a peritonitis, an inflammation, infection of the peritoneal membrane. All right. So as we already went over the subdivisions of the large intestines in that, from that picture, right? And we're not going to worry about all the bends and flexures along the way. I didn't make you guys memorize that in lab. We're not going to get into all of that detail, right? So this is, again, all of these areas of the large intestines, right? By the time the material reaches the rectum, we should have absorbed water, most of the water from it, leaving just a little bit in there. And this should be, again, where all the remaining waste products, bacteria, mucus, and material that we can't digest are building up. And when the rectum gets enlarged with this fecal matter, it can trigger again the urge to defecate, a bowel movement, right? So again, we mentioned about those fatty appendages that are on there that don't seem to have a really any significant role in any physiology. Tende coli, again, it's an additional band of muscle in the large intestines that again creates those hostra pouches along the large intestinal structures, right? So these hostra pouches are going to help with the mixing of our food. So the remnants of our small intestine. So as we're pushing that fluid through here, these ridges and pouches in there are going to churn that material that's still fairly liquidy to make sure we're absorbing water from there and evenly drying out and creating a more compact fecal material. Mass movements, powerful peristaltic movements along here are going to move food through this area and help create that fecal material. And this is where your diet can affect some of this, right? So if I'm eating fiber-rich food or taking fiber supplements, that undigestible fiber material as it reaches the intestines, and again is material the bacteria can be ingesting, it's going to create a more powerful movement of the material through the digestive system so that I have more regular bowel movements, right, and less chances of developing constipation. But if I'm not eating a very fiber-rich diet, those chances that I won't have enough of this movement of the large intestines, and I might dry the contents of my intestinal fecal material too much and create a harder fecal matter that is harder to push out during a bowel movement. Also with bacterial infections, right? So if I get a bacterial diarrhea, right, that could increase intestinal movement of the intestines and help flush that infection out of the intestines. But this is also why things like Imodium AD, while in some cases may be good to stop diarrhea, but if it's caused by an infection of a certain bacteria, trapping the bacteria in the intestines and decreasing the motility of the intestines would actually in some cases maybe create more of a problem, because diarrhea is a natural defense to try to push the infection out of the intestines and clear the body of that foodborne illness, because most of the time these would be coming in from contaminated food into the body. Right, so large intestine is going to lack any villi because they're not really involved in a lot of absorption, right? A lot of gland activity predominating in mucus production to help again lubricate the intestinal wall as this fecal matter gets pushed through there to make sure again we can move this material, especially as it gets more compact and harder, through the intestinal area. Not any significant enzymes produced in here because we're not really doing any nutrient digestion. That should have all happened from the duodenum and passing through the small intestines. All right, so here's looking at the histology of the large intestines. Right, as we move to the anal canal, right, the anal canal again has anal columns in there. Again, this is where you're getting your hemorrhoids, the veins in this area, as they get distended and stretched out, can create difficulty having a bowel movement, because as the fecal matter passes, that enlarged lump along the anal canal will create some discomfort. Right? So here are these little anal canals. Again, we got all these blood vessels. You can get what's, again, that hemorrhoid forming in there. So here's our, again, internal sphincter of the anal canal, smooth muscle, involuntary. External sphincter of the anal canal, skeletal muscle, so voluntary. So that anal sphincter, the external anal sphincter, is what we learned to control when we went from being a baby to a toddler, right? And we didn't need to wear diapers anymore. Again, at what point someone needs to stop wearing diapers will vary from age to age from individual. But once you learn to control that muscle, you didn't have accidents, and you could, again, go to the have a bowel movement only when you were ready and you're sitting on the toilet in the bathroom, right? Smooth muscle of the internal sphincter is just autonomic control. So that's what, again, predominated when you were a baby. Food got to the intestines as the pressure built up, stimulated the relaxing of that muscle. You had a bowel movement, no external skeletal muscle control to prevent that bowel movement. And that's also something as we get older from age, and other issues may cause a lack of control of the external sphincter, and you might not be able to control your bowel movements. More often people have urinary incontinence problems, which we'll talk about as they get older. Not as often anal issues with not being able to control bowel movements. But again, some people can have that issue as well, especially if they're in long-term care facilities and have various health issues. Right, so we've already talked about those sphincters, right? The anus is the exiting area. That, again, is now going to be keratinized epithelial tissue, right? So the intestines had more of a columnar epithelium. As we transition to the anus, again, that becomes skin tissue and becomes keratinized epithelium, right? Probably a little higher up, it's going to be non-keratinized stratified squamous, but as we get into the anal canal, it becomes keratinized epithelial tissue, right? So again, the large intestines accounts for about 10% of your nutrient absorption. As we said, B vitamin 5, vitamin K, biotin, which is also B vitamin, can be produced by bacteria absorbed in there. The last bit of water, about a liter of water is absorbed through here, again, which is, again, only the last bit of water, because a large amount of water would be absorbed in the small intestines, right? So water reabsorption, 1.5 liters of water enters the colon over 1 liter of water reabsorbed, right? 200 mLs of water remains in the feces as it's lost from the body. If you have more water in there, you might have diarrhea, which could lead to dehydration, especially people who have severe diarrhea. Can, again, cause problems with dehydrating because they're losing more water from the body and not absorbing as much of it, so they're not hydrating themselves as completely as they can, right? So about 200 mLs remains in your feces, right? 70% of the water, right, is 20% is undigestible material, inorganic material, right, and bacteria, right? So a large amount of bacteria in your fecal matter, along with, again, byproducts of bacteria, ammonium, indole, hydrogen sulfide, which is why, again, your bowel movements isn't a pleasant smelling aroma to it, because of all these products of bacteria associated with it. And if you have a diarrhea and you're pushing food through there too quickly, again, that can intensify some of these smells, and a large amount of bacteria from higher up in the intestines are coming out along with that are creating that not so pleasant smelling diarrhea. Defecation involves long reflexes, central nervous system, right? Short reflexes, again, involving that myoenteric plexus that we talked about earlier, right? So once again, a large amount of food, the remnants of our food, the undigestible fecal material gets to the rectum. Short reflexes stimulate smooth muscles, right? Long reflex, again, with the spinal cord, controls the skeletal muscles that, again, we can control that bowel movement within reason, right? That's why when you have foodborne illness, diarrhea, you can sometimes have diarrhea you can't control because it's just so much stimulation of the rectum trying to control that. With the skeletal muscles becomes impossible, and you'll just have bowel movement whether you're ready to or not. So we'll stop here, right, and get ready for the last section of the system.